In this video, we will explore the complex condition known as diabetic cardiomyopathy. We will examine its causes, mechanisms, and clinical implications, including the role of lipid accumulation, autophagy, mitochondrial dysfunction, and inflammation. We will also discuss the connection between nephropathy and cardiomyopathy and touch on potential therapeutic targets. Let's begin. Diabetic cardiomyopathy is associated with increased lipid accumulation in the myocardium, a phenomenon known as cardiac lipotoxicity. Excessive levels of free fatty acids and triglycerides can disrupt myocardial metabolism, leading to impaired energy production, mitochondrial dysfunction, and cellular toxicity. Lipotoxicity also promotes inflammation, oxidative stress, and apoptosis, further contributing to the development and progression of diabetic cardiomyopathy. Understanding the mechanisms underlying cardiac lipotoxicity is crucial for identifying potential therapeutic targets for managing this condition effectively. Autophagy is a cellular process that helps maintain cellular homeostasis by removing damaged or dysfunctional organelles and proteins. Impaired autophagy has been implicated in the pathogenesis of diabetic cardiomyopathy. Dysfunction in the autophagic machinery leads to the accumulation of damaged organelles, protein aggregates, and oxidative stress, exacerbating cardiac dysfunction. Elucidating the role of autophagy dysregulation in diabetic cardiomyopathy could provide insights into therapeutic interventions modulating this process for cardioprotection. Mitochondria play a crucial role in myocardial energy production, calcium handling, and redox signaling. In diabetic cardiomyopathy, there is a disruption in mitochondrial structure and function. Increased oxidative stress, impaired mitochondrial biogenesis, altered fusion-fission dynamics, and defective mitophagy contribute to mitochondrial dysfunction. This impaired mitochondrial function leads to energetic deficits, impaired calcium handling, increased apoptosis, and ultimately, cardiac dysfunction. Unraveling the mechanisms and consequences of mitochondrial dysfunction is vital for developing targeted therapies to manage diabetic cardiomyopathy effectively. In the video, we will explore the role of oxidative stress in diabetic cardiomyopathy. The excessive production of reactive oxygen species and the imbalance with antioxidant defense mechanisms lead to oxidative damage in the heart, causing inflammation, fibrosis, apoptosis, and remodeling. Understanding redox signaling pathways and finding therapeutic targets are crucial in managing this condition. Advanced glycation end products, AGEs, are formed through a non-enzymatic reaction between reducing sugars and amino acids. In diabetes mellitus, chronic hyperglycemia leads to increased accumulation of AGEs, contributing to cardiac remodeling and functional impairment. AGEs induce inflammation, fibrosis, and cross-linking of extracellular matrix proteins, resulting in increased myocardial stiffness, diastolic dysfunction, and impaired ventricular contractility. Exploring the mechanisms underlying AGE-mediated cardiac remodeling can help identify potential therapeutic targets to prevent or reverse the detrimental effects of AGEs in diabetic cardiomyopathy. In diabetic cardiomyopathy, the renin-angiotensin system, RAS, is dysregulated, leading to increased levels of angiotensin II. This promotes inflammation, oxidative stress, fibrosis, and cardiac dysfunction. Understanding the RAS in diabetes can help identify potential therapies to restore cardiac function. Diabetic cardiomyopathy is commonly associated with type 2 diabetes, however, it can also be observed in individuals with type 1 diabetes mellitus. Although the fundamental pathological processes in type 1 and type 2 diabetes differ, both conditions share some common mechanisms contributing to cardiac dysfunction. Understanding the unique features and underlying mechanisms of diabetic heart disease in individuals with type 1 diabetes is crucial for tailoring management strategies and improving outcomes in this specific patient population. Diabetes often leads to microvascular disease, which impairs blood flow and damages the inner lining of blood vessels. In diabetic cardiomyopathy, this compromised blood supply causes reduced oxygen and nutrients delivery to the heart, leading to inflammation, fibrosis, and dysfunction. Understanding this link can help develop new treatments for protecting the heart. Diabetes mellitus often leads to the simultaneous presence of both cardiac and renal complications, namely diabetic cardiomyopathy and diabetic nephropathy. Both conditions share common pathogenic mechanisms, including oxidative stress, inflammation, fibrosis, and endothelial dysfunction. The interplay between these two complications can potentiate the development and progression of each other, resulting in worse outcomes and increased cardiovascular risk in patients with diabetics. Understanding the shared mechanisms and clinical implications of such comorbidities is essential for comprehensive patient management and tailored therapeutic strategies. Chronic low-grade inflammation in type 2 diabetes contributes to diabetic heart disease. Pro-inflammatory cytokines like IL-1 beta, TNF-alpha, and IL-6 promote cardiac damage. Understanding their role can help develop anti-inflammatory treatments for diabetic cardiomyopathy. Diabetes mellitus is associated with an increased risk of both cardiovascular and neurological complications. While traditionally studied separately, emerging evidence suggests shared pathogenic mechanisms and interactions between diabetic cardiomyopathy and diabetic neuropathy. Oxidative stress, inflammation, mitochondrial dysfunction, and microvascular disease contribute to the development and progression of both cardiac and neurological complications in diabetes. Understanding the shared pathways and interactions can provide valuable insights for designing comprehensive management strategies targeting both cardiac and neurological manifestations in individuals with diabetes mellitus. Despite ongoing research, therapeutic options for diabetic cardiomyopathy remain limited. However, emerging research has identified several potential molecular targets that may be exploited for developing novel therapies. These targets include AMP-activated protein kinase, peroxisome proliferator activated receptor agonists, sirtuins, cardiac microRNAs, and stem cell-based therapies. Investigating these novel therapeutic targets may pave the way for the development of personalized interventions to improve cardiac function and prevent the progression of diabetic cardiomyopathy. In conclusion, diabetic cardiomyopathy is a multifaceted condition with significant implications for diabetes patients. Understanding the intricate mechanisms involved can lead to individualized treatment strategies and improved quality of life through translational research and therapeutic targets.